Every day, an intricate battle unfolds within our bodies. A battle between the vigilant cells of our immune system and potential threats, including cancer cells. But how exactly does our immune system recognize and eliminate these rogue cells? Let's delve into the science behind this crucial defense mechanism. The immune system is a complex network of organs, tissues, and cells working in harmony to protect us from disease. It comprises two main branches: the innate immune system, which provides immediate but non-specific defense, and the adaptive immune system, which mounts a delayed but highly specific response. For example, natural killer cells, or NK cells, are critical components of the innate immune system. They respond rapidly to infected or transformed cells without prior sensitization. In contrast, cytotoxic T lymphocytes, or cytotoxic T cells, are key players in the adaptive immune system. They require antigen presentation and activation to specifically target and destroy cancer cells that present tumor antigens. Both NK cells and cytotoxic T cells are essential for immune surveillance and the elimination of malignantly transformed cells. Cancer cells originate from normal cells that have accumulated genetic mutations due to factors like environmental carcinogens, radiation, or inherited genetic defects. These mutations can activate oncogenes or inactivate tumor suppressor genes, leading to uncontrolled cell proliferation. Mutations often result in the production of abnormal proteins, sometimes known as neoantigens. Neoantigens are novel peptide sequences presented on the cell surface by specialized protein complexes called MHC molecules, or major histocompatibility complex molecules. Because they are not present in normal cells, neoantigens can be recognized as non-self by the immune system, prompting an immune response specifically against cancer cells. In effect, the immune system considers cancer neoantigens to be foreign. Central to immune recognition is the major histocompatibility complex, or MHC. MHC molecules are like display platforms on cell surfaces, presenting peptide fragments to immune cells. There are two classes of MHC molecules: Class 1 and Class 2. MHC. Class 1 molecules present endogenous peptides. Those peptides generated within the cell. These small peptides are processed from normal proteins or mutated proteins such as neoantigens. Peptides on MHC class 1 molecules are presented to CD8 positive cytotoxic T cells. The MHC class 1 antigen processing pathway involves proteasomal degradation of intracellular proteins, peptide transport into the endoplasmic reticulum via TAP proteins, and loading onto MHC class 1 molecules. This allows cytotoxic T cells to monitor the health of all nucleated cells and detect abnormalities such as viral infections or oncogenic transformations. Conversely, MHC class 2 molecules present exogenous peptides to the immune system. In other words, peptides that are derived and processed from external sources. Peptides on MHC class 2 molecules are presented to a different type of T cell, known as a helper T cell. The function of helper T cells, as their name suggests, is to provide help to other T cells. For example, through the production of cytokines, helper T cells provide stimulatory signals that activate cytotoxic T cells. Cytotoxic T cells possess T cell receptors, or TCRs, that recognize specific antigens presented by MHC class I molecules. The TCRs are generated through random genetic rearrangements, creating a vast repertoire of T-cell receptors that are capable of recognizing a multitude of different peptide antigens. When a TCR binds to a neoantigen peptide MHC complex, it triggers signal transduction pathways, leading to T-cell activation. Co-stimulatory signals, such as CD28 on the T-cell, binds to CD80 and CD86 molecules on professional antigen-presenting cells, such as dendritic cells. These co-stimulatory signals are required to fully activate the T-cell. Activated cytotoxic T-cells then proliferate and differentiate into effector cells capable of killing target cells. Upon recognition, the cytotoxic T-cell releases cytotoxic granules containing two important molecules, perforin and granzymes. Perforin creates pores in the target cell's membrane, facilitating the entry of granzymes. Once inside the target cell, granzymes initiate apoptosis by activating caspases and inducing DNA fragmentation. The initiation of apoptosis, or programmed cell death, causes the death of the cancer cell. Additionally, cytotoxic T cells can express a cell surface molecule called Fas ligand, which binds to Fas receptors on target cells. This binding then triggers the extrinsic apoptotic pathway, thereby killing the target cancer cell. Cancer cells often attempt to evade cytotoxic T cells by reducing MHC class I expression on their surface, a process called MHC downregulation. NK cells can detect this loss through a mechanism known as missing self recognition. NK cells express a balance of activating receptors like NKG2D and inhibitory receptors such as KIRs and CD94 NKG2A. Inhibitory receptors recognize self-MHC class I molecules, sending inhibitory signals to prevent NK cell activation. When MHC class I molecules are absent or reduced, the inhibitory signals are diminished, allowing activating signals to dominate. Activated NK cells eliminate target cells through several mechanisms, for example through the release of cytotoxic granules containing perforin and granzymes. This mechanism of killing is similar to cytotoxic T cells, thereby inducing cancer cell apoptosis. NK cells also express the death receptor ligands, TRAIL and Fas ligand, which bind to death receptors on target cells, triggering apoptotic pathways. In addition, NK cells can participate in an important immune process known as antibody-dependent cellular cytotoxicity, or ADCC. NK cells express CD16, also known as Fc gamma receptor 3, which binds to antibodies coating the cancer cells. This leads to NK cell activation and the release of perforin and granzyme, leading to target cell destruction. Cancer cells employ sophisticated strategies to evade immune detection and destruction. Cancer cells secrete TGF-beta, IL-10, and vascular endothelial growth factor, which suppress effector immune cells and promote the development of immunosuppressive cell types. Regulatory T cells, or Tregs, are recruited by tumours. Tregs suppress cytotoxic T cell activity through cell contact mechanisms and the secretion of inhibitory cytokines such as TGF-beta and IL-10. Myeloid-derived suppressor cells can further inhibit T cell activation and promote tumour growth. Tumour cells can also increase their expression of checkpoint molecules such as PD ligand 1 or CTLA4 ligands. By upregulating PD ligand 1 or CTLA4 ligands, cancer cells engage inhibitory receptors on T cells, leading to an suppressive or exhausted T cell phenotype. To counteract immune evasion, researchers have developed immunotherapies that enhance the immune system's ability to fight cancer. For example, checkpoint inhibitors are designed to release the immune system's brakes. Anti-CTLA-4 antibodies block CTLA-4-mediated inhibition during T-cell priming in lymph nodes, enhancing T-cell activation. Anti-PD-1 and anti-PD ligand-1 antibodies act in peripheral tissues, reinvigorating exhausted T cells within the tumor microenvironment. Checkpoint inhibitors are beginning to have some success in treating various forms of cancer, such as melanoma or breast cancer. CRT cells, or chimeric antigen receptor T cells, are engineered to recognize specific tumor antigens, such as CD19, in certain B-cell leukemias and lymphomas. The CAR construct combines an antigen binding domain, usually derived from a monoclonal antibody, with intracellular signaling domains that provide activation and co-stimulatory signals, bypassing the need for conventional antigen processing and presentation. Personalized cancer vaccines are being developed by identifying patient-specific neoantigens through tumor sequencing. Identifying neoantigens involves whole exome sequencing of tumor and normal tissue to detect somatic mutations. Bioinformatic algorithms are then used to predict which mutated peptides can bind to the patient's MHC molecules with high affinity, thereby maximizing the chances of mounting a vigorous cytotoxic T-cell response. Dendritic cells can be loaded with these neoantigens to effectively present them to T cells. This approach is known as a dendritic cell tumor vaccine. Alternatively, synthetic long peptides can be used to stimulate robust CD4 positive helper T cell and CD8 positive cytotoxic T cell responses, thereby stimulating the immune system to mount an anti-tumor response. Advancements in immunology and biotechnology are paving the way for more effective cancer treatments. Future directions include combination therapies, which integrate immunotherapies with chemotherapy, radiation, or targeted therapies to enhance antigen release and modulate the tumor microenvironment. Identifying genetic or immunological biomarkers that predict patient responses to immunotherapy could enhance the effectiveness of treatment modalities, while exploring how the gut microbiota affects systemic immunity and responses to cancer therapy could further aid treatment strategies. Neoantigen load and immunogenicity is also important. This involves a better understanding of how the quantity and quality of neoantigens influence immune responses and tailoring therapies accordingly. Synthetic biology approaches, such as engineering immune cells with enhanced functionalities, such as resistance to immunosuppression or the ability to secrete therapeutic molecules, is another potential avenue of research that could have positive benefits for cancer patients. The immune system's ability to recognize and eliminate cancer cells is a testament to its sophistication and adaptability. By unraveling these intricate mechanisms, scientists are developing innovative therapies that harness our own immune system to combat cancer. Thank you for joining us on this exploration of the immune system's fight against cancer. We hope this deep dive has illuminated the incredible complexity and potential of immunology in oncology. Together, let's continue to unravel the mysteries of the human body